So today you are going to study about fluid mosaic model of plasma membrane. In short form we'll call it as PM. Fluid mosaic model of plasma membrane. Okay. So who gave this model first? Singer and Nicholson in 1972. These two scientists gave this model of plasma membrane. What is this model explaining us about? This model is explaining about cell membrane. We also call it as plasma membrane. So we are studying the structure of cell membrane by using fluid mosaic model, which was given by Singer and Nicholson in 1972. So students, if this is the cell now, okay, animal cell doesn't have any cell wall, so directly it starts from plasma membrane. Okay, so outside like this, they will be arranged in this format. Both outer side and inner side you can see the structures like this. So whole membrane is covered, cell is covered by plasma membrane. Okay, in detail we will see the structure now. Okay, so if you see here, you can see two layers here. So your plasma membrane is made up of two layers of what? Phospholipid. So we call it as phospholipid bilayer. Nothing but they are made up of phospholipid bilayers. So here you can see one outside layer is there, one inside, like this. They are arranged outside and inside. Okay, their arrangement is called as amphipathic. Why we call it as amphipathic arrangement? So if you see the both structure here, okay, they are having like this head and tail region. Okay, both the layer you can see head and tail region. Now I'll tell you why we call it as amphipathic. Okay students, see the head here. Both the sides, head is towards outer side. Okay, outer side of the membrane. Here head will be arranged here like this, outer side. Here also tail you see here, they are towards inner side. They are somewhat protected towards inner side. The tail is like this, towards inner side in the cell. So you can see here, coming to the structure, head is towards outer side, tail towards inner side. Tail is protected. Why I'll tell now. This head, they are polar. Okay, head is polar, means they are water loving, hydrophilic. Okay, head region is water loving. They are made up of phosphate and glycerol. So they are loving the water. They are water loving. They love water. H2O. But now tail region, they are protected inner side. Can see here now, tail is towards inner side. They are water hating. They don't like water. Now they are mainly made up of lipid. Lipid don't like water. If we put some oil and all in the water, it will not get dissolved. Same way, lipid don't like water. So they are towards inner side. So that tail region we call it as hydrophobic. Phobia means afraid. The tail region is afraid of water, so we call it as hydrophobic. Let's write here tail as hydrophobic. Tail is hydrophobic. Head is hydrophilic. Such arrangement, okay, head towards outer side, tail towards inner side, tail towards inner side, head towards outer side, they are called as amphipathic arrangement. So we call phospholipid bilayer as amphipathic arrangement. Okay, now next, what you can see here in blue colors, you can see here, they are proteins. Now when I tell proteins, there are two types of proteins. Okay, you can see this larger ones throughout the membrane. They are called as intrinsic proteins. Proteins which are present embedded inside the membrane. you can see here outside some proteins. Here you can see they are extrinsic proteins, proteins which are present outside the membrane. So two types of proteins are intrinsic protein and extrinsic protein. And now you are studying about our RBC cells. Okay, this is the plasma membrane structure in our RBC cell. So when it comes to animal cell, cholesterol will be also present. Okay, cholesterol will be present, and you can see here, okay, sugars. Sugar will be present in the plasma membrane. Either they can be branched like this, they are branched, or they are just unbranched. So sugar can be branched or unbranched also. Okay. Now I'll tell you the composition. Okay, proteins are present 52% in our plasma membrane. Lipids 40%, carbohydrates 2 to 5%. Carbohydrates are very less, that is sugar. Cholesterol is also present in animal cell when it comes to animal cell. Okay, so just let us revise now. Today you have learned the structure of cell membrane or plasma membrane. This structure, this model is called as fluid mosaic model. It was given by Singer and Nicholson in 1972. They are semi-fluid structures. Why? Because this lipid, they have fluidity. They give fluidity to the structure. It's like, say, in the ocean. Okay, now lipid is the ocean. Think. In this ocean of lipid, your protein is floating like this. How? As a iceberg. How iceberg will float? Same way, lipid is having fluid nature. This protein will be floating in that ocean of lipid. So we get semi-fluid nature. Fluid nature is for lipid, and little rigidity will be given by proteins. So this structure is having lipid bilayer, which is made up of phospholipid, and two types of protein, intrinsic and extrinsic. Now you know what is intrinsic. Intrinsic means which is present embedded inside the membrane. Extrinsic just outside the membrane. Now functions of plasma membrane. It helps in transportation of material. Your plasma membrane will help in transportation of material. There will be small pores through which material can go out, come inside. They are also called as a semi-permeable membrane, and we can also call it as differentially permeable membrane, because this plasma membrane will not allow all the molecules to come out and come in. Only particular and only small molecules can come out and go inside. So it also helps in locomotion. Amoeba, pseudopodia. This amoeba will produce like this pseudopodia structure, which helps in locomotion, movement. They protect the animal cell from injury. It forms like a jacket around the animal cell. This plasma membrane is forming like a jacket like this. So helps in protection. Okay, students. Did you all understand today's class?